Hello, and welcome to the first ever virtual Mile No-Till Day. My name is Kendall Sheldon, and I'm a master's student working under Dr. Shekoofa at the West Tennessee Research and Education Center in Jackson, Tennessee. Today, I want to talk to y'all about cover crops and their negative allelopathic impacts. I'm sure you're all familiar with cover crops and their benefits to a field, including water infiltration, soil health, and weed suppression. But allelopathy may be a new term, so allow me to explain it. Allelopathy is simply the ability of one plant to inhibit the germination and/or growth of another through the production of chemical compounds. A few ways that these allelochemicals can become present in the soil are from leaching from leaves, exudation from roots, and decomposition of plant litter. So now that you know what allelopathy is, let's move on to see what kind of impacts it can have. Here are two pictures of alfalfa fields in Georgia. The alfalfa on the left was planted into a field with no history of rye. There are some notable weeds, but overall the stand and growth of the alfalfa looks good. Now the field on the right is the same cultivar of alfalfa, but it was planted following rye used as a cover crop. Although weed suppression is evident in the field, the stand and growth of the alfalfa is poor. Looking at these pictures side by side, you can see the negative allelopathic impact that the cover crop rye had on not only the weeds, but also the main crop in that field, which was the alfalfa. This brings us to the question, can different cover crops cause diverse allelopathic effects on goosegrass and soybean germination? If you're wondering why goosegrass and soybean were both chose for this study, it relates to the two alfalfa fields in Georgia. It's important to know how cover crop allelochemicals impact weeds as well as the main crop for this study, which is soybeans. So in 2019, 7 cover crop species were selected: wheat, cereal rye, hairy vetch, woolly pod vetch, crimson clover, winter pea, and canola. The two timings were long season and cover crop species planted following corn. The termination timing was the 3rd week of April in 2019. Middle Tennessee and East Tennessee were the 2 locations chosen, and 2 biological reps were sampled from each location. Here is a picture of the cover crop field in East Tennessee roughly a month before harvest. At the top of this slide is our secondary location in Middle Tennessee. Now I want to walk you through the process of taking these cover crops and turning them into an extract. Beginning with harvesting, these cover crop samples were hand clipped in the field, and then that fresh plant material was dried in an oven for 5 days at 60 degrees Celsius. Then that dry plant matter was ground in a mill to create a powder. That powder was then mixed with deionized water and centrifuged to remove any solids from the liquid. Then the liquid was filtered, and that created our pure extracts. From those pure cover crop extracts, a treatment was created with a 1-to-1 ratio of the cover crop extract and deionized water, and water was a secondary treatment. This study was split into 2 experiments to become more manageable, one that focused on soybean and the other that focused on goosegrass. The treatments were applied to the seeds every other day for 7 days. Germination was recorded daily in order to calculate germination percentage and germination rate. At the end of the 7 days, root and shoot lengths were measured for all of the seeds. Now I'm going to walk you through the results. For the soybean root length, we have 2 graphs, one for East Tennessee and one for Middle Tennessee. The cover crop extracts reduced root length compared to water for all cover crops, but for East Tennessee, winter pea had the highest impact, and for Middle Tennessee, wheat had the highest impact on soybean root length. For soybean germination percentage, both locations had similar results, and compared to water, all cover crop extracts did reduce germination, but canola had a significant impact on suppressing germination. Now moving on to goosegrass. For goosegrass root length, hairy vetch had the highest impact on reducing root length for both East Tennessee and Middle Tennessee locations. And for East Tennessee, wheat was also a cover crop that had a high impact on reducing root length, whereas Middle Tennessee also had winter pea that had a high impact on reducing root length. Goosegrass germination percentage. For East Tennessee, all extracts did reduce germination percentage, but wheat was the significant extract that had the highest impact on germination percentage. In Middle Tennessee, woolly pod vetch, canola, and winter pea had the highest impact on suppressing goosegrass germination. For the overall soybean results, canola showed the greatest allelopathic effects on soybean germination. Looking at the picture at the bottom of the slide, there are two treatments, the water treatment on the left and the canola extract treatment on the right. You can visually see that the water-treated soybean seeds have a much higher germination percentage and overall root length compared to the picture on the right, which represents the canola extract seeds. Most cover crop extracts significantly reduced seedling root length, and almost all cover crop extracts significantly reduced germination rate. For the overall goosegrass results, in East Tennessee, crimson clover and wheat, and in Middle Tennessee, woolly pod vetch and winter pea significantly reduced the goosegrass germination percentage, while it had no significant impact on soybean. This means that these cover crops from these locations suppressed goosegrass germination while having no negative effect on soybeans. Therefore, it would be more beneficial for a farmer to choose one of these cover crops to plant ahead of soybean versus canola, whose extract had negative impacts on soybean. What I want to leave y'all with today are 3 take-home points. It's important to know the cover crop species before planting soybean. The location and environment may change cover crops' allelopathic potential, and goosegrass seed germination and root length were suppressed with cover crop extracts application. This shows that due to their allelopathic properties, cover crops could be used as a new method for weed suppression, which would be beneficial as herbicide resistance becomes more common. I would like to thank the Tennessee Soybean Promotion Board for supporting this research, and I'd also like to thank all of you who are at home watching. I know this isn't the no-till day that anyone expected. If you have any questions, the contact email is below. Please reach out and we will be happy to answer any questions you may have.